Warm welcome to this wonderful session, Chapter 13, Part 3. In this part, we have to discuss cyclic photophosphorylation. Then we have to see 𝕆𝕆𝕋𝔼𝔼 hypothesis, in which we have to discuss how ATP are generated in photosynthesis. Then we will switch on to dark reaction, and a part of dark reaction, that is Calvin cycle. Detail of this we have to see today. This is Part 3, Chapter 13, that is Photosynthesis, Part 3.
Already in Part 2, we have discussed about non-cyclic photophosphorylation. That is, in non-cyclic photophosphorylation, you have seen that the electron which comes out from photosystem second, it will go to the electron transport system, then it will go to photosystem 1, and after photosystem 1, it will be received by, that is NADP, means the electron will not be returned to the original site. Therefore, that was called as non-cyclic photophosphorylation. Because this movement of electron will generate ATP, that's why this is photophosphorylation. Detail of this we will discuss right now. The second one is cyclic photophosphorylation. As the name indicates, here that the light will be trapped by photosystem. This is photosystem 1. The photon will excite the reaction center of this, that is P700. Electron will jump, will be accepted by electron acceptor, and then it will go to the electron transport system. And after generating, that is hydrogen ion, again that will be back to the photosystem 1, the same chlorophyll molecule, therefore known as cyclic photophosphorylation. You see, the electron is circulated within the photosystem, and photophosphorylation occurred due to cyclic flow of electron. Therefore, that was known as cyclic photophosphorylation. The possible location where this could happen is in 𝕆𝕆𝔼𝕆, while the membrane or lamellae of grana have both PS1 and PS2. It's very important to understand. You see, children, that let this is the granum. Let this is that is granum we have here like this, and let this is that is 𝕆𝕆𝔼𝕆. So this cyclic photophosphorylation we have here in 𝕆𝕆𝔼𝕆. PS1 is here, while in granum lamellae we have both PS1, that is on outer side, and PS2 on inner side. This I told you, excited electron doesn't pass on to NADP in this cyclic photophosphorylation. That will be back to the photosystem 1 complex through electron transport chain, and therefore that is cyclic flow will result, and that is only the synthesis of ATP will be here in cyclic flow. There is no reduction of NADP, and there is no generation of oxygen, or oxygen is not liberated. There is no photolysis of water, is it? Now we can compare the cyclic photophosphorylation and non-cyclic photophosphorylation. We may summarize once. What is cyclic photophosphorylation? This is PS1. Electron will jump, will go to the electron transport system, will be back to the same, and here through electron transport system it will generate, that is hydrogen ions, which will in turn will give, that is ATP. While in non-cyclic one, that is PS2, electron will go here, will go to the electron transport system, will be back to, that is photosystem 1, again will go uphill movement, then that will be accepted here by NADP, which will receive this hydrogen ions and will reduce this NADP to, that is NADPH. Again we have summarized it. Now it's easy to differentiate. Cyclic photophosphorylation, only photosystem 1 is involved. It involves both PS1 and PS2. Movement of electron here is cyclic, will be returned to the original site. Here the movement of electron is non-cyclic, it is unidirectional, will not be back to the same site. Only ATP are generated here. In this case, both ATP and NADP will be formed. Oxygen is not evolved. There is no photolysis of water. Oxygen is evolved, and as a result of which the electron liberated by photolysis of water, they will go to the, that is to the chlorophyll molecule, which has giving out its electron. It is dominant in photosynthetic bacteria. It is found in higher plants. Photolysis of water, as I told you, it is not here in this. The photolysis of water takes place here. Now the important thing here is, which is a very, very important one. You see, in electron transport system, when the electron will be transported through electron transport system, they will be from high energy level towards that is low energy level. One thing happens here in electron transport system, that the protons will be released, and these protons ultimately will get accumulated in the lumen of grana. That is the granum, and this one is that is lumen of granum. And this accumulation of proton here in grana, that is here the concentration will be very, very high. Now these protons, again they will like to release into stroma, and this release cannot be through any place. There is a particular site is there, and that site is known as ATP synthetase enzyme. Through this, these protons from high concentration to low concentration will be released in stroma, and here that is they will generate the ATP. This is the basic feature of hypothesis, chemoosmotic hypothesis. That is how the ATP are generated in photosynthesis. That was proposed by 𝕆𝕋𝔼𝔼 to explain mechanism of ATP synthesis. This, now we are talking about chloroplast. Almost similar process is there in mitochondria. Here ATP synthesis in chloroplast is linked with the development of that is proton gradient across the membrane of thylakoid. This is thylakoid membrane, and here see, this is the proton gradient. In the lumen the concentration is very, very high. This concentration in lumen may be about 1000 to 2000 times more than the concentration of protons in stroma. This is known as that is proton gradient, that is gradient, right? There is one difference, that is here the proton accumulation is towards the inside of the membrane, that is in lumen of the, that is in chloroplast, that will be lumen of the chloroplast, while in case of mitochondria you have to study in time to come. In mitochondria that will be in between the two membrane, that this is the outer membrane, that this is the inner membrane, the proton concentration that will accumulate in between the two membranes. Now we have to see, it's a very interesting one, that what's the reason that proton gradient will develop, or concentration of these protons will be much more in grana lumen as compared to stroma. There are three reasons. One which is very important one, this is photolysis of water. You know, splitting of water is very much essential in photosystem second, and the photosystem second, you know, is located on inner side of thylakoid, that is on oppressed part. The protons or hydrogen ions which are produced by photolysis of water, that is splitting of water, they will get stored there in lumen of the thylakoid and will increase hydrogen ion concentration. You see in this diagram we have here, yes, that is, you see, this is the diagram, this is the grana, this is the lumen of the grana, this is the membrane, outer membrane this one is, and this one is, that is the inner membrane, and this is the lumen. Towards the inner side of the, that is thylakoid membrane, this is photosystem second, what I told you right now. You see, that is here photolysis of water will take place. As a result of this photolysis of water, oxygen will be released which will go out. The hydrogen ions will be released which will get accumulate here in lumen, and the electron that will go to the photosystem second, the chlorophyll molecule which has lost the electron will become normal again. Is it very much clear? So one reason was, that was photolysis of water. Photolysis of water will take place here. The second important reason here is that is known as 𝕆𝕍𝑃 pump. 𝕆𝕍𝑃 pump. You have to see what we mean by. You see, children, here from photosystem, that is second, it is here, the electron will go, that will be received by electron acceptor, which is lying towards outer side of the membrane. That this is the thylakoid we have here. It is located towards outer side of the thylakoid membrane. Now this electron that will go to this is 𝕆𝕍𝑃. From 𝕆𝕍𝑃 the electron want to go to the electron transport system. Here, that is, it will accept the hydrogen, so that hydrogen ions and the electron, they will become hydrogen. And now when this electron will be passed on to the electron transport system, that is, these protons will be released here. So that is known as 𝕆𝕍𝑃 pump. You see here in this diagram, from photosystem, this is pheophytin. It has gone to the 𝕆𝕍𝑃. 𝕆𝕍𝑃, that is, electron will be transferred to the cytochrome system, but it will not receive the electron, it will receive the hydrogen. This hydrogen ion, it is there in, that is, stroma. Therefore, this 𝕆𝕍𝑃 on one side will take these ions from, that is, stroma, and then it will pump these ions to the lumen of chloroplast. That is known as 𝕆𝕍𝑃 pump. The third important one here is, see again here, that is, you see here, this is PS second. The electron has gone here. It has gone to the electron transport system. Here, this is the 𝕆𝕍𝑃 pump, what I told you. See here, that is, in this, that protons have been released, accumulate here because of photolysis of water. Because of 𝕆𝕍𝑃 pump, that the protons have been again accumulated here in this. After that, what happens? The electron from electron transport system will go to the photosystem 1. From photosystem 1, they will go here and ultimately will be received by, that is, NADP. This NADP will receive protons from, that is, stroma again. So when the protons will be consumed through this process, there will be decrease in, that is, concentration of hydrogen ion. Decrease, therefore, a gradient will be maintained because of this also. Here in lumen, the concentration of that will be more comparatively. So there are the three reasons. One is, that is, photolysis of water. Second is 𝕆𝕍𝑃 pump. And third is, what is this third one? This is, that is, in reduction of, yes, reduction of NADP, that is, use of proton ions. Is it very much clear? We have to see little detail of this. That is, one, I told you about photolysis of water. What is transfer of hydrogen ions from stroma to lumen? That is related to 𝕆𝕍𝑃 pump. We can see detail of this. Electron move through photosynthesis. Proton are transported across the membrane. This happens because primary acceptor of the electron, which is located towards outer side of the membrane, you must be getting what I am saying here. They will transfer its electron not to electron carrier but to hydrogen carrier. Is it? Hence the molecules removes a proton from stroma and will pump this, that is, proton towards lumen of the, that is, thylakoid. The third one, right now I told you detail, reduction of NADP, that is, towards stroma. It will consume proton ions from stroma, which is also responsible for, that is, difference in proton concentration or the developed proton gradient. Is it right? Now you see, what happens now? You see, in chloroplast, proton in the stroma decreases. That is reason number 3. And because protons have been used up by, that is, NADP in its reduction, and this creates a proton gradient, what I told you. This gradient is important because the breakdown of this gradient that lead to synthesis of ATP. Very important point we have here. You see first what will be here, children, that is in chloroplast lumen we have that is high concentration of proton. They will like to move from high concentration to that is low concentration. Low concentration of protons we have there in that is stroma. These protons cannot come out into stroma, that is through the membrane of chloroplast, because this is impermeable. But in membrane of the chloroplast we have that is ATP synthetase enzyme. It's this very part that is lies in the membrane. That part is known as CF0, and the part which is projected in stroma, that is called as CF1. These hydrogen ions, they will be passed on to that is this ATP synthetase, and here the configurational changes will be there which will generate ATP. Is it right? The gradient is broken down in that way due to the movement of a proton across the membrane and through this transmembrane channel, that is this is CF0 of ATP synthetase. ATP synthesase enzyme consists of two parts, what I told you. One is that is CF0, which is embedded in the thylakoid membrane and form transmembrane channel through which the protons from lumen will like to come to that is stroma. It will facilitate diffusion of proton across the membrane. The other portion is that is CF1. It protrudes into the outer surface of the thylakoid membrane towards stroma. Right? What happens now? The breakdown of gradient provides enough energy which will bring out configurational changes, conformational changes in CF1 particle and will generate ultimately that is energy, which is energy is released that will be packed and will be stored there in ATP. Chemoosmosis requires a membrane. Important. What is required? It requires a membrane of thylakoid, it requires a proton pump, it requires a proton gradient, and it requires that is ATP synthetase enzyme. Is it clear? Energy is used to pump proton across a membrane to create gradient or high concentration of a proton within the thylakoid lumen that I told you. ATP synthetase, yes, has a channel that allows diffusion of proton back, that is across the membrane, because rest of the part of the chloroplast membrane, or better to call that is granum membrane, that is impermeable, and it will activate ATP synthetase enzyme movement of protons which will catalyze the formation of ATP. Is it right? Along with NADPS produced by movement of electron, the ATP will be used immediately in biosynthetic, that is reactions which will take place in stroma, because this ATP which have been released here, which have been produced in light reaction, and NADPH which have been produced, both of these are known as that is assimilatory power, and this assimilatory power produced herein, that is light reaction, that will be used there in dark reaction. Is it right? Now you see, before we proceed, you have to see that what we mean by this diagram. Again you have to see here that this is the diagram here. This is the ATP synthetase enzyme. This part that is known as F0, better to call it at, we may call it CF0, chloroplast F0, and that will be known as CF1. This is the transmembrane channel which is embedded in thylakoid membrane, and this is protrude towards the stroma. That is ATP synthetase enzyme, that the energy generated here will be used, that is in formation of ATP, that is high energy, that is molecule, that is from ADP and inorganic phosphate. Is it very much clear to you? Okay. Now we have to proceed to the that is remaining part of this, that is in photosynthesis you have seen, that is in light reaction, children, you have been told in light reaction, which have been used out of the that is 4 requirements of photosynthesis. Light is essential here, chlorophyll is essential because it will trap the energy, and water is essential in photolysis of water. As a result of light reaction, what you have seen, what will be formed, that is this ATP will be generated, and here this is NADPH NADPH will be generated. Now they will be used therein, that is dark reaction. Dark reaction. Why it is known as dark reaction? This is a misnomer term. Dark reaction, that is actually we mean that this will happen in dark, but you have to remember nothing happens there in dark. It is known as dark reaction because it doesn't need light directly. It is independent to light. Till the product formed in light reaction, ATP and NADPH are available, the dark reaction will continue because both of these will be consumed in dark reaction in CO2 fixation, and again they will be back, they will be charged in light reaction. Is it clear? We have to see now dark reaction. What is dark reaction? That is ATP and NADPH to produce in light reaction. What I told you right now. Are you getting my point? You have to concentrate. They are used to drive, that is process leading to the synthesis of food, that is carbon dioxide assimilation. This dark reaction, this is biosynthetic phase, that is different type of enzymes will be used here. This process doesn't need, that is light directly. It is independent to light, isn't dependent on light directly. Only the product of light reactions are being used, that is still ATP and NADPH are there. Dark reaction will continue. It will, that is fix the carbon dioxide. Therefore, to call this phase dark reaction is misnomer. The site of dark reaction, that is stroma in chloroplast. And now we have to say that depending, very important point this is, in dark reaction, depending upon the first stable product of carbon dioxide fixation, there are, that is two main pathways. One is C3 pathway, also known as that is Calvin cycle, because the detail of this that was traced out by Calvin and co-workers. And why this is known as C3? Because in this pathway, stable product after CO2 fixation, that has 3 carbon, and that one is phosphoglyceric aldehyde. On other side, C4 pathway, detail of this was traced out by, that is H and SLEC, therefore known as H-SLEC cycle. And similarly, the first stable product in this, it has 4 carbon, and that one is oxaloacetic acid. Is it clear? Now we have to say first the Calvin cycle, C3 cycle, also known as reductive pentose pathway. Some important point related to this. Calvin and his co-worker, that is Bansham and Basham, in 1949, they work out the whole pathway of carbon dioxide assimilation, and they use unicellular alga. One is, that is chlorella, one is Cnidesmus, and they showed the pathway operated in a cyclic manner, the RUBP that was generated. To find out the pathway of carbon dioxide fixation, they used radioactive isotope of carbon, that is C14, and the techniques chromatography and radiography. Important this is. C3 cycle, first stable product, as I told you, that has 3 carbon, and that one is phosphoglycerate or phosphoglycerol aldehyde. This is one very important point we have here, that is Calvin pathway is found in all photosynthetic plant. Either this is C3 plant, in C3 plant it will have definitely C3 cycle, and in C4 plant, you see in C4 plant it is also having C4 cycle followed by, that is C3 cycle. So C3 cycle is very important, it is found in all. And the Calvin cycle, that is C3 cycle, we can divide into 3 important steps. One is carboxylation, one is reduction, one is regeneration. Carboxylation means here the fixation of carbon dioxide. This is, that is received, that is fixation of, that is carbon dioxide. Then the product will be reduced, phosphoglyceric acid which is being produced, that will be reduced, that is into phosphoglyceric aldehyde. And then regeneration, carbon dioxide acceptor, which is, that is RUBP, ribulose biphosphate. It is to be regenerated, which acts as, that is CO2 that is acceptor. Is it clear? We can see, children, now that is the cyclic way. That is 3 important steps we have. This is the one that is carboxylation. This is RUBP. This one is which acts as CO2 acceptor. It will receive the carbon dioxide, that is as a result of which 3-phosphoglycerate will be formed. By using this ATP and NADPH, that will be reduced. That is the second step is reduction. As a result of reduction, that will be formed, that is triose phosphate will be formed, that this is phosphoglyceric, that is aldehyde, 3-phosphoglyceric aldehyde. A part of this will be utilized in making food, that is glucose, which will be polymerized to give starch, and that is maximum part of this will be used in regenerating, that is again this CO2 acceptor. That one is, that is CO2 acceptor. In that way, a cyclic process starts, regeneration that consumes, that is ATP. So this is part 3, chapter 13, photosynthesis. Thank you.